So, continuing chapter 6. Figured I'd show pseudopods. Amoebas, asexual reproducers, so these probably are all genetically identical. They've all got the little identifying badge things that hello, my name is, and there's a bunch of different names on them. It just says amoeba conventions. Spelled the old way, amoeba, with an O in the middle of it. They've been trying to get that O out of there for a long, long time. While I've got free space here, this is the internal structure of a cilium or a flagellum. Remember, they're homologous, so that they've got this microtubule core in what's called, you don't need to know the 9+2 arrangement, but I'm going to tell you about it anyway, in what's called the 9+2 arrangement. And it grows from underneath the surface and then grows out in a projection. There's a scanning electron micrograph of a ciliated protozoan with a band of cilia and a bunch of cilia, and it actually is grabbing something and pulling it into a food vacuole, endocytosis. Just to show you, lots and lots and lots of little cilia. He told you that? Well, he's pulling your flagellum, Nancy. And there's a ciliated protozoan up there with a bunch of cilia. Okay, now we're going to go inside the cells. Starting with, when people started to be able to look at cells under a microscope, for various reasons, occasionally the cells will break open and the contents will spill out. But what spills out is fairly thick, kind of jellyish, kind of holds together for a while before it kind of disperses. So they called it cell jelly, which is literally what cytoplasm means. And that is the juice inside the cell. But of course it's water and a whole bunch of other things. Is enough that if you put it into fresh water, there's always going to be more water headed in than can be headed out, because there's just so much other stuff in there as well. There are structures in there that give cells their shape, that hold things in position. When you look at your cheek cells under the microscope, they pretty much always have the same shape. That's why when you draw me a picture, I know whether you're looking at the cheek cell or not. And the nucleus is there, and it has this particular shape, and it's in a particular spot in the cell because it's held in place by cables and struts and connectors and such. And at some point, when people started to figure this out, it said, okay, it's like a skeletal structure inside the cell. So we'll call that the cytoskeleton, because they're used to cyto meaning cell. But nobody wanted to go to the Latin dictionary and look up what Latin for skeleton was. And this is just holding some of the things in position that we're going to be talking about as we go through the bits and pieces that are inside a cell. It gets fairly complicated. Lots and lots of stuff in there, cables and struts, and these are 2 rows of cilia also, with microtubules sticking out through there. Part of the cytoskeleton are membranes inside the cell. But they also have these other things. Little bitty tubes, like the ones that go up the middle of a flagellum or a cilium. And so they went, well, they're little bitty tubes, and they kind of knew micro. So rather than look up the Latin word for tube, they called them microtubules. Eventually they found more things that were solid strands, thinner than microtubules, and they called them microfilaments. There's a third group that kind of falls in between. They call intermediate filaments. We're not going to worry about those. But that's the components of the cytoskeleton. Now, inside the cell are little bits and pieces that do particular jobs. And at some point, somebody went, well, it's like organs in a body, right? Yeah. Well, then let's call them organs something. They didn't call them microorgans, they could have. They called them organelles. We talked about this way, way, way back when as part of the organization of cells. And organelles fall into a couple of categories. Is that we're going to start with organelles that are just collections of molecules. There's a bunch of these, but we're only going to talk about 3 of them. One of them you've already heard of. Everybody's heard of chromosomes. Turns out, in living cells under a microscope with the light coming through the right way, when they're dividing and you can see the chromosomes, they tend to be shiny and have like colors. So they were called colored bodies chromosomes. And before they show up as individual bits, they show up as strands, and they called that chromatin. And then later they found out that's always there. It's just, with a light microscope, you can't see it till it gets somewhat packed up. And so this gets a little confusing where chromatin is going to mean one thing when we're watching cells divide, and it's going to mean something else when we start talking about how chromosomes work. It's the material that chromosomes are made of, and it's a combination of the DNA, right? DNA, the genetics, and proteins that give the chromosome structure. And a chromosome is a long strand of DNA with a whole bunch of different genes on it. Genes are codes for proteins, and those need to be made eventually. Proteins are made in the ribosomes. Chromosomes are inside the nucleus, ribosomes are outside the nucleus. But they're involved in, the code goes out and then it's fed in. We'll talk about the steps later. But ribosomes are a little cluster of RNA and proteins, and the code feeds through and a strand of amino acids comes out, and it's going to be your proteins. One other unfortunate. A structure that's in the nucleus that's a molecular complex. Somebody decided to call it a nucleolus or a nucleolus. There's a couple of different ways to pronounce this, but it's so similar to the word nucleus that it's way more confusing than it should be. Well, we're stuck with it, like we are with a lot of terms. And this is a little clump inside a nucleus. Sometimes there's just one, sometimes there's a bunch of them, and anything in between. And this is where the RNA gets dealt with. The RNA is processed there, pieces of RNA are stored there. Is that RNA is a big player, remember, in how nuclei work. And so it's got like a little repository, processor of RNA. And some nuclei need them in a few different places, so there'd be more than one nucleolus. Nucleoli would be the plural. And some of them just really only have one. Okay, those are the molecular complex organelles. The other category are membrane-based. And I'm going to throw a picture up here before I write any more than this. We will eventually go through pretty much all of these. Not quite, but close. And this is the nucleus. This is a nucleolus inside the nucleus. And these little dark dots are ribosomes. Now, this particular picture has the ribosomes all stuck in these membranes. This is endoplasmic reticulum. We'll talk about that later. But ribosomes also often are free-floating in the cytoplasm. It all depends on what you're making with them and what you want to do with it. So this picture, I think they just didn't want to make it too confusing, so they didn't put dots all over the place. But, so you've got your ribosomes, you've got your nucleolus, and let's see, trying to find, yeah, there's no chromosome showing. I was drawing a blank as to what the third molecular complex organelle was. Now we're going to do membrane-based organelles. Little bubbles that are called vesicles or vacuoles, Golgi complex where secretions are packaged, endoplasmic reticulum, which can have ribosomes embedded in it and be rough, or not have it and be smooth. I'm going to list all of these eventually. Here's the outer membrane. There's mitochondria that do aerobic respiration. Centrioles are the one thing we're not going to talk about. That looks like a little vesicle. Lysosome. Lysosome is a type of vesicle. And that's what we're going to start with. We're going to start with the simple membrane-based organelles that are basically bubbles. You got this big chamber of chemicals. And you often need a chamber to put stuff in temporarily or to do something special away from the rest of the cell. The same way the cell could be isolated from other stuff around it, the chambers can be isolated inside the cell to do special chemical stuff. And when they're little, they're called vesicles. When they're big, they're called vacuoles. Like cilia and flagella, there's no one cutoff point where anything smaller is a vesicle and anything bigger is a vacuole. Is that there are big vesicles and little vacuoles, but typically these are smaller and these are bigger. And there are some that have their own special names that are very common and have their own special functions. So we're going to list a few. It is by no means the whole list. It's just a little partial list of these. So this includes lysosomes, lyse from the word for breaking or digesting. These guys move digestive enzymes from one place to another inside the cell. They're inactive enzymes until they put them where they need to activate them, but they get them from one place to another. And there's a similar type of little bubble called peroxisomes. They have peroxide in them a lot. And lysosomes often are digesting stuff that's been brought in from outside the cell. Peroxisomes tend to recycle stuff inside the cell that you don't need anymore, but you would like the bits and pieces to do stuff with. So peroxisomes are internal recycling for the most part. Lysosomes are outside digestion, bring it in, let's digest it. And they work together with a couple of other parts of the cell to do a particular function that I'm going to list here. So this is not an organelle, this is a process. This is sometimes called cell suicide, where a cell will purposely kill itself. It's worn out, it can't do what it's supposed to do, it's getting out of the way of other cells. This is something that happens in multi-celled systems. It happens when you develop, that's what happens to the webbing between your fingers, is those cells all died to give you wiggleable separate fingers. And a lot of times this is something that goes wrong when you get cancer, where a cell should kill itself because it's really screwed up. But instead of killing itself, it just starts dividing out of control and becomes a cancerous growth. But I know this looks like apoptosis. This is the way a lot of people pronounce it. I just remember reading an article a few years ago saying this part is Greek and that P is silent, so it's actually apoptosis. But it's biology. If you go to a conference and there are biologists talking there, somebody's presenting, you'll be sitting there and people go, what word was that? What word was that? And and somebody writes it out and they go, wow, I've never heard anybody pronounce it that way before. If there's 5 different ways to pronounce a word, you'll find all 5 pronunciations if you wander around and listen to people. So, it's biologists are very flexible in that sort of stuff. As long as we can figure out what you're saying, we're generally okay. Other types of vacuoles. So the outer membrane folds up around something that needs to be taken into the cell. But once it's in there, it needs to be broken down for the cell to be able to use it. So that bubble that's come in where the membrane folded around something and then brought the bubble in, that is a food vacuole. So it's a bubble with food in it. In plants, in small green plants, the carbohydrates that they use in their cell walls are not that strong to hold the plant up. They have another way to help hold the plant up, and that is they put a bunch of particles into a big vacuole in the middle of every cell. Water goes in, pushes out on the vacuole, pushes out on the cell, stiffens everything like blowing up a balloon. And that gives the plant more stiffness, holds it up better. Which is why if you haven't watered your houseplants in a while, they start to wilt, they start to droop. They start using that water for photosynthesis, and they lose the pressure inside the central vacuole. And I talked about this before, where you take small single-celled organisms and you put them in fresh water, and they don't have a casing to protect them to keep them from getting too big, and they're not waterproof because they can't interact with their environment if they're waterproof. So they have to pump water back out, and that's done with a contractile vacuole. Fill it, empty it, fill it, empty it, fill it, empty it. Now this would typically be way bigger than the lysosomes, but we're stuck with what they drew. So food gets surrounded by a bubble, the bubble goes in, you need to break the food down, here comes a lysosome, it fuses with the 2 bubbles together, the digestive enzymes get activated and you get digestion. This is to a large extent what a peroxisome would do. They've changed the names of these since this was made. So this is an example of a peroxisome, but that isn't what they're calling it. Because it's recycling part of the cell itself. This is taken into the peroxisome and it's broken down for recycling. Plant cell with this big chamber inside that's pushing out on the cell walls. The cell walls are not as stiff as you would like in order to hold up the plant. This only works for green stemmed plants, as woody plants do have enough carbohydrates to hold themselves up. But this is your central vacuole in the middle of the cell. And everything kind of gets crowded off to the edges. There's your nucleus and your nucleolus and your endoplasmic reticulum with the ribosomes and Golgi bodies and mitochondria, chloroplasts, which we haven't talked about yet, weren't in the animal cell, but they're in the plant cells. And these would be microtubule struts, reinforcing, holding everything kind of in place. This is an electron micrograph, but somebody colorized it. And this is a cell showing the central vacuole and the nucleus and some other structures. These are chloroplasts. This is a living single-celled animal taken with a fancy microscope so you can see structures inside better. And this is a pretty advanced one. It's got 2 contractile vacuoles with little canals that bring water in. And it fills these up and then they squirt the water out. And so these are your contractile vacuoles of a protist. Remember protista? Golgi apparatus is the next thing, so before I write that down, Golgi apparatus are flattened stacks of bubbles, and stuff comes in from the rest of the cell and this is going to make things that are going to leave the cell, secretions, right? Cells secrete things. They secrete hormones, they secrete neurotransmitters, they secrete toxins, they secrete all sorts of stuff. And it's got to be made somewhere. And what is good about a Golgi body is it makes the stuff, and then these little bubbles pinch off from the ends. You can see them pinching off there. This is an electron micrograph. And these flow up to the surface, become part of the cell membrane, and then what was in there is now outside of the cell. Now, one of the confusing things about this is that some books call them bodies, some books call them complex. That overhead called them apparatus. It always is Golgi. In some cases, when it has this many names, people will actually tell you that Golgi bodies are different from a Golgi complex or different from Golgi apparatus, but I've never really run into that. It's just that they can't quite decide what English word they're going to stick on this. But again, here's your stacks. Here's what they call secretory vesicles, pinching off, being carried up to the surface. There's a surface over there. And then the secretions wind up outside of the cell. The cell walls on plant cells are partly made with materials that are produced in Golgi apparatus. And this is the next thing we're going to talk about. That's your canal system to move stuff around inside a cell. And it's inside the juice, and it's a network. Endoplasmic, inside the juice, reticulum from a Latin word for net, and it's canals where, okay, I'm gonna make something and I'm making it this place in the cell, but I need it to go somewhere else. Or I need it to maybe to go 5 different places. And so it's kind of like a highway system for liquids. And it carries stuff from one place to another in the cell. It kind of works its way through the juice. There are other ways to move stuff around a cell. Microtubules actually work like little conveyor belts. You clip stuff on and away they go. But this is a major network inside cells. Smooth endoplasmic reticulum is literally just the network. But think of the highway. What do we got around here? We have all these distribution centers where stuff gets, well, not made. Used to be factories. But if you want to move stuff on the highway, then you need someplace nearby that's going to make the stuff or at least get the stuff and distribute it. And so rough endoplasmic reticulum, which was in all those other pictures, embedded in the membrane are the ribosomes. So the proteins are made right on the edge of the highway, and off they go from where they're made to wherever they're going to get used. You don't always need that. You don't always need ribosomes on your network. So sometimes it's just just the network, and sometimes it's production in the network as well. And this stuff very often is very much associated with the nucleus, because the nucleus is where the codes are. These are what make the proteins from the codes. So the nucleus is where your DNA is. It's where your chromosomes are. That's where your DNA is processed. It's where the production to make proteins starts. It doesn't end there. It goes out into the cytoplasm for the stuff to be made in the ribosomes. But your nucleus is a big store separate spot for your DNA and RNA processing. And it turns out that when you see it in the cell, it looks like it's got a membrane around it. It turns out it's got a 2-layer membrane around it. So instead of calling it a nuclear membrane, they call it a nuclear envelope to acknowledge that it's not just a single layer. Two more. I'm just going to write these down. Where are we standing time-wise? Oh, we're too far in. This one's going to be a long video, sorry. Too close to the end to just put it on another one, though. So you get these chambers where aerobic respiration happens or where photosynthesis happens. And back 50 years ago, Lynn Margulis, a researcher who eventually wound up in Massachusetts, I don't know where she was when she did this, but she was interested in how this aerobic respiration worked in mitochondria compared to bacteria that do it, because bacteria also do aerobic respiration. And so she started to study the bacteria, and what she found were these guys look just like the bacteria. And when she started to look at the chemistry that these guys use matches the chemistry that the bacteria use. And so she came up with a theory that these two organelles did not just spontaneously evolve inside big eukaryotic cells, that they were originally specialized bacteria, specialized prokaryotes that were taken in for food, but they were more useful alive than digested. And so the cells that held onto them and took advantage of the processes they could do had a huge advantage over cells that just digested them. This is billions of years ago. And when the cells divided, they made sure that these guys divided inside the cells and got distributed. And so it started as an arrangement where the prokaryotes that were taken in were protected and fed. And then they did all this great stuff: aerobic respiration, very efficient energy system; photosynthesis, very efficient energy system. And eventually it got to the point where they no longer could live on their own. They didn't really act like bacteria completely anymore. And people looked at this theory and went, "Oh, Lynn, go away. Yeah, this is nuts. Yeah, they're doing the same thing. Of course they look the same way and they're using the same chemistry." And they were very resistant to the idea. It was a very big new idea that very often the powers that be are resistant to that. It probably had something to do with the fact that she was a woman and it was the 1960s, and the old guard was old men. Still is to some extent. And they weren't ready to listen to a big radical idea from a woman. But here's the kicker. What she found in this thing and this thing, there's little prokaryote chromosomes in there, little loop chromosomes with some specialty genes that work for mitochondria and specialty genes that work for chloroplasts. You can't build a whole mitochondrion out of it because some of the stuff to build this, those genes are now in the nucleus. It does a better job of processing than these guys do. But stuff very much specialized for these two guys, they have remnants of those original bacterial chromosomes. It was kind of hard to argue with it at that point. So people went, yeah, okay, Lynn, we buy it. And now it's a pretty well accepted idea. That is the chapter on cell structures.